All right, if you came in late, grab a handout on the other side. It's the lecture exemption study guide. Grab a handout. All right, we're going to have 2 lectures on lecture exam 3 material, then 1 week from today it is lecture exam 2. So let's look at lecture exam 3 material, the study guide. Remember how small lecture exam 2 study guide was? This one's 9 pages. Basically, lecture exam 3 is going to be about nervous system. Things about the nervous system. So we'll do some preliminary stuff this week on the nervous system. And then we'll look at the central nervous system, which is the spinal cord and brain. We'll start on the spinal cord and move up to the brain. There on page 4 and 5. I will show you how to organize content on the brain, and I will tell you right now, if you plan on taking physiology, it would be very advantageous to pay good attention to that. You'll be at a pretty severe advantage having this cover the brain for your anatomy, because it's pretty similar in physiology, maybe 20% more. Then we do the peripheral nervous system. Then we do look a little bit more in detail with the autonomic nervous system and how to whiteboard there. Then we go into senses. We'll spend most of the time on the senses on the eye and the ear. We do some of the minor senses. And hold on, let's see, depth, Anderson at the back. Yeah, and then we'll finish it with the endocrine system. Oftentimes students at this level of anatomy and physiology consider the endocrine system different from the nervous system because there are different chapters in the textbook. But in reality, in medicine, the endocrine system is the same as the nervous system. They are 2 ends of the same spectrum, which is why neuroendocrinology is a specialty in medicine. Okay? I tend to teach things more on a medical level than I do as some of the textbook authors like to organize. Okay. Any questions on the lecture exam 3 study guide? A little bit more hefty than lecture exam 2 in it. A little bit 2 more pages for lecture exam 4. Okay. Let's first of all look at the organization of the nervous system. These are anatomical divisions. So we have the overall nervous system. First we can identify the brain and spinal cord as being the central nervous system, or the CNS, central nervous system. And the PNS is just everything else. Okay? We're going to go into more details on each one of these divisions later, so don't laugh at my artwork, but that's supposed to be brain and spine. In the peripheral nervous system, we can identify parts of the peripheral nervous system that deal with things you're conscious of, that's somatic, and then we will deal with things that you are not conscious of, that is autonomic. Be careful with spelling. It's not automatic, it is autonomic. And then within the autonomic division, we can see some nerves that are involved in the fight or flight responses, sympathetic, and then some having to do with ease and feast responses, that's parasympathetic. But those 2 terms only apply to the autonomic division. And this is pretty ubiquitous. You can go online, you can see these same figures. Okay? Just different levels of structural organization of the nervous system. Are we all clear? Now let's go in a little bit more in detail on each one of these levels. First, difference between central nervous system and peripheral nervous system. Central nervous system is the brain and spinal cord. The peripheral nervous system is everything else. You don't have to write this down, but I want you to hear it. These distinctions here on this slide are gross anatomical distinctions. They are not neurological distinctions. They are gross anatomical distinctions. Gross anatomical means I can take out the cadaver, boom. Put this finger on the CNS, boom. Put this finger on the PNS. That's gross anatomical distinctions. Everybody got it? What that means? It's not a neurological distinction because if you look at these blue neurons, and then what I drew here in colors are neurons, part of it is in the CNS and other parts of it are in the PNS. Everybody got it? So the same neuron can be in both. It's not a neurological distinction. It is a gross anatomical distinction. Okay? Now there are also some differences in terminology here. So go ahead and fill in this line. So I don't assume you guys know what these terms mean. When it says fibers arranged in tracts in the CNS, that's where they're talking about this long process called an axon. We'll talk about this more in just a bit, but I want to make sure you guys understand what these terms are. The nerve cell bodies, where the nucleus and other organelles are, if there's a collection of them in the CNS, we call it a nucleus. And then sometimes the axon is covered with a layer, and if it is covered with a layer, it is myelinated. And the myelination is caused by a neuroglial cell. Remember, when we did the lab quiz way back in week 3. In the nervous system, you had to know there are 2 types of cells: neurons that carry the nerve impulses, and the neuroglia cells. And the neuroglia cells help neurons function better. Does this ring a bell? Well, these are neuroglial cells. And when they form a covering over the axon, it's myelinated. While the bottom bullet is in the CNS, the neuroglial cell is called an oligodendrocyte, and in the PNS, the myelinating neuroglial cell is neurolemmacyte. Everybody got it? Does this help? Having these drawings up here?
Do those neurofilaments only cover the axon terminal, or do they cover any?
Oh, they'll cover the whole axon.
Okay.
I just don't want to draw it all out.
Okay.
Okay. But there are small gaps in between. You see the blue? There are small gaps in between. That's important. Everybody got this? Let me give you a, well, I'll talk about this in just a bit. So far, so good? Here, remember that CNS and PNS is a gross anatomical distinction. You can actually put your finger on it. So here in blue, CNS, in yellow, PNS. Everybody got it? Now let's talk about some things we can identify out in the PNS, and that is the somatic subdivision and the autonomic subdivision. In a nutshell, somatic things are things you are conscious of. Autonomic things are things you are not conscious of. And this is both motor and sensory.
Like you know you're voluntarily doing it, or you're aware of it? You're just
If you're touched and you're aware of it, that is somatic sensory. And on the motor side, there's only one thing that you can consciously do motor-wise, and that's contract skeletal muscle. Okay? You don't kill the one. Okay, now things that you're conscious of are both motor and sensory, and things that you are not conscious of are also motor and sensory. For things you're conscious of, if you're touched or if you're moving, contracting skeletal muscle, there are these sensory receptors in and around your muscles that you know your muscles contracting and moving. Those are all things you're conscious of. It's somatic sensory. There's only one thing on somatic motor, and that's skeletal muscle, like this. Only thing, skeletal muscle. For the autonomic, you have smooth muscle and cardiac muscle, or even glands and other organs, like in your digestive tract. That's all under autonomic control. And they can both be excited or inhibited, whereas skeletal muscle can only be excited. Okay? So in that box, it kind of shows you a list of autonomic motor. Can anybody give me an example of autonomic sensory? Sensory that you are not aware of.
Your heart.
Okay, that's motor.
Oh.
Anybody?
Blood flowing?
What's that?
Blood flow.
Blood flowing, well that's from the heart, and that's still motor. Talking sensory.
Breathing.
What's that?
Breathing.
Breathing is under autonomic control, but that is motor. Yes?
Like how your body ignores the sensation of your clothes on you?
Yeah, that is actually still somatic sensory, but it has to do with sensory receptors showing sensory congregation. That's a little bit different. Anybody else?
Smells.
What's that?
Smelling.
No, if your lab partner farts, you're aware of it. That's somatic. Yes?
You can't see your eyelashes? Like your brain kind of puts them out?
Yeah, that's true. We're barking up the wrong trees here. And you know why? Because you're not conscious of it.
When is your hair growing?
Yeah, that's still motor, but because it's things that are happening out. When we're talking sensory, we're talking action potentials, these nerve impulses going into the brain. Motor, it's going out of the brain. So sensory, we're talking about signals going into the brain, but you are not conscious of it. Shall I finally just break the ice and tell you some examples?
Yes.
Do you know what your blood pH is? Your brain knows. It's getting autonomic sensory, your blood pH. How about partial pressure of CO2? You know what it is? Your brain does, autonomic sensory. How about blood glucose levels? Your brain knows. It's getting autonomic sensory. You're just not conscious of it. Is this, finally, now you know what things that you're conscious of and things that you're not conscious of. Does this help? There's a whole realm you are not conscious of, but we know about. Okay. All right, now let's talk about the location of nerve cell bodies. Everybody know what a nerve cell body is now? Where the nucleus and organelles are? Over here, this is all multipolar neurons I've drawn here. The little eyelashes are dendrites. The circle is the cell body. And then the long line is the axon. Does that help?
Yeah.
Here, if you notice, for the somatic, all the circles are in the CNS. There are no cell bodies outside of the PNS. That's what it's saying right there.
What happens to
There's only 2 neurons, and both of the cell bodies are in the CNS. But on the autonomic side, you have these two same thing in the CNS, but then you got this green one that's out in the PNS. And a collection of cell bodies in the PNS is called a what? Collection of cell bodies in the PNS is called a what?
ganglion? Oh. So 2 is a collection? 2 points is a collection?
Well, I'm only drawing it
Oh, it's
schematically.
more?
It is a whole bunch.
Okay.
So it looks like is there more than one in the ganglion?
Yes,
is.
there would be a whole bunch.
Okay.
It would be a collection. And it would be what color of ganglion?
Gray.
Gray. Wherever you have a collection of these guys in the CNS,
Oh, that's why it makes it gray.
it would be gray. But we would call these collections in the CNS what? Look on that same slide.
Nucleus.
Nucleus. Everybody, are you connecting the dots here?
Yeah.
Apply the words. This is ASL, anatomy as a second language. So in this box here, it says we have neurons that have cell bodies in the CNS. The blue one in the CNS is the preganglionic neuron. Then they synapse with postganglionic neurons whose cell bodies are in the ganglion and the PNS, the green one. All right, everybody look at me. You can be passive at words, or you can take the words and draw this out, and now you know what these words mean. This is active learning. This is what I did as a student, as I was sitting in your chair. And the guy didn't have anything on any screens, because that's how old I am. He's just talking, referencing figures in the textbooks. We all had our textbooks open. He's just talking. Well, what was I doing? I took my Nerd 4-color pen and I was drawing this out. Everybody got it? Any questions?
When they form up the ganglion, do they just get larger to go into the postganglionic, or do they actually change their, do they actually change?
The neuron itself.
Yeah, because it's going from preganglionic to ganglion and then postganglionic. Are they just moving, or are they actually changing?
No, the only thing that is moving with these arrows, that's the direction of the action potential, the nerve impulse.
Okay.
Because that's why it's motor. It's going out. In fact, I want to bring these terms to your attention right now, so I'll just leave it right here. If this is motor that I'm drawing on the whiteboard, you can see why in the CNS you will use the term descending. The action potential is going down from the brain down to the spinal cord. Everybody got it? Then, once it's going out in the PMS, it's going out, and we call that efferent.
Efferent?
Efferent, right up there in the middle whiteboard.
Oh, efferent.
Now, if you're having sensory from your skin, somatic sensory, these signals are going afferently in. Then it's going to go ascending up to your brain. Do you see these terms?
Mm-hmm.
These terms will even help you in English, by the way. You know that you have an effect on others, but other people's effect to you.
That was so hard for me to try of those two. Yeah.
That's it. It's tied right to this.
Yeah.
Everybody got it? By the way, your goal when you take English 101 is not to pass English 101. Your goal is to learn how to write at a college level. Everybody got it? It's not about getting a grade, because there's plenty of teachers at this college, I'm afraid, that you can get a passing grade in English 101 and still not write at a 10th grade level. That doesn't do you any good, even if you have a passing grade in your transcripts. Because then you've got to write lab reports for me, and you don't even know how to proofread. And even 10th graders know you got to proofread your stuff before you turn it in. I get people just typing things out like it's a text. Turn it in. That's not college level. That's your goal. Take English 101 to write at a college level, not just to get a passing grade. Because it's embarrassing. Okay. Let's talk about sympathetic and parasympathetic. Again, these are summary comments, but sympathetic has to do with fight or flight responses. Again, you can't control this. You cannot consciously control it. And parasympathetic has to do with ease and peace responses.
What's peace?
I'll explain this. Fill in the blanks, and let me explain this later. Again, this is just an introduction to these concepts. Later on, we'll go into this in more detail. Okay, first row is the summary. That's up on the bullet above it. Second row where it says origin. Just to the left of this slide where it says origin, you want to write where the preganglionic axon comes out of the CNS. Origin means where does the preganglionic neuron originate? Where the preganglionic neuron, the blue one, comes out of the CNS. So let's talk about that. You guys remember the sections of the vertebral column? Cervical, thoracic, lumbar, sacral, coccyx. Everybody remember that?
Yes.
What this second row is saying is that the sympathetic preganglionic neurons, axons, come out in the middle. Thoracolumbar's in the middle. For parasympathetic, it comes out in 4 pairs of cranial nerves on top and 3 pairs of sacral nerves on the bottom. Sympathetic comes out in the middle, parasympathetic comes out top and bottom. What I've drawn on the whiteboard would be sympathetic, because it's coming out in the middle. Everybody got it? If it was parasympathetic, it would either come out of here or come out of here. If it's coming out in the middle, it's sympathetic. Third row. I would never even bother to memorize something like the third row. Because anatomy and physiology is about you. All I would do is remember what it's like to be a human being. And from my own personal experience, I know what it's like to have a fight or flight response. And I know what it's like when I come home and mom's cooking Thanksgiving dinner. That would be the ease and feast response. Your mouth starts watering. Your stomach starts growling. Ease and feast response. Coming home for Thanksgiving, fight or flight response. We're going to see one of those movies that'll probably come out this month. Everybody got it? You can figure it out if you are self-aware. You don't need to study it for a test. Just be self-aware. This class is about you. All right, fourth and last row has to do with the neurotransmitters released at the effectors. So since I wrote effector up here, that's what's talking about what's coming out of this axon terminal. If it's sympathetic, it's norepinephrine. If it's parasympathetic, it's acetylcholine. That's what's coming out of the green one. Can anybody tell me what the end of this blue line is? It relates to the lecture exam you're studying for. What is the end of the blue line up on top going biceps brachii?
Neurotransmitter plates.
Getting warmer. The end of the blue line is the motor end plate at the neuromuscular junction.
That's what I said.
Blue plus black equals the neuromuscular junction. Right here. This is the neuromuscular. You guys remember the sliding filament theory essay?
Yeah.
Step 1, got the neuromuscular junction. You guys remember what's released here?
ACH.
It's also acetylcholine,
I can't put it.
right?
Oh.
Acetylcholine.
Just trying to tie things together, even though it's another lecture to have, it's one body. Okay. Any questions? All right, now if you notice here, first there's the nervous CNS in the middle. You can see parasympathetic comes out, top and bottom. Sympathetic comes out in the middle, thoracolumbar. Everybody see that? Notice the organs on both the left and the right side are the same organs. Do you see that? This is referred to as dual innervation. It's getting both sympathetic and parasympathetic. Everybody look at me. I want you to plant this seed right now, and this will make things a lot easier if you just get this through your heads. Even though we're talking about CNS and PNS, and somatic and autonomic and sympathetic and parasympathetic, it makes everything sound black and white. There is no black and white in your bodies. It is all shades of gray. And because these organs are getting both sympathetic and parasympathetic, in dual innervation, the way your body functions is on ratios, all shades of gray. It's not all one or the other, it's both. But which one's higher? Sympathetic to parasympathetic ratios. Everybody got it? Just get that through your heads now. Because when things are taught at this introductory level, it makes things sound black and white, and there are no black and whites. It's all shades of gray. Everybody got it? The fourth way, and this is where we're going to talk about some of those terms, is a functional organization, and that is whether it's sensory or motor. In the PNS, they're called afferent or efferent, and the CNS, we use ascending and descending. And that is through all levels of the previous slides. See the sensory motor. That took a long time. All right. Everybody got that? Okay. Let's see if this rings a bell. You guys remember how skeletal muscle fibers need to have insulation around them? Let's see if you guys recognize these prefixes. Now we're talking about a nerve trunk out in the PNS. There's connective tissue around the entire nerve trunk called epineurium. What is it called around the entire muscle?
Epimysium.
Epimysium. Does this ring a bell? Axons tend to run in bundles, and we actually use the same name as we use for a bundle of skeletal muscle fibers, which is what? Fascicles. And surrounding an entire fascicle of axons is perineurium. What do we call it around a fascicle of skeletal muscle fibers? Perimysium. See the parallel? Then around individual axons is endoneurium. And surrounding individual muscle fibers is endomysium. The reason why it's parallel is because both muscle fibers and axons carry action potentials, and you've got to insulate both. They're very parallel, and so we use parallel prefixes. Epi, peri, and endo. Okay. Everybody got it?
What's it say in the white over that bottom slide?
Thin layers of endoneurium around axons. Fibroblast, nucleus. Yeah, we have a saying for these TVs.
Shit.
Low bid. College is going to pay whichever is the lowest bid. All right. Now let's talk about neurons. And this is what is drawn over there in blue, over there on the right. The cell body, also referred to as soma, includes a nucleus and little packets of rough endoplasmic reticulum and ribosomes we call chromatophilic substance. Old name is Nissl bodies. And Nissl bodies are bluish gray in color. And when the cell body is full of these bluish gray packets, the whole cell body becomes bluish gray, and that's what makes gray matter gray. It's the cell body. Dendrites are where a neuron can get incoming signals. They can also get incoming signals on the cell body. So even though it says that on the slide, yeah, you need to know both of these as the input zone. Not just the dendrites, also cell body can get incoming signals. Then in this one, this is a multipolar neuron, you have one axon. The base of the axon is called the axon hillock. This is important for physiology. When you take physiology to understand where the axons are. It's just the base of the axon as it comes out of the cell body. Now there's always in a multipolar neuron only one of these, but once that axon leaves the cell body, it can branch dozens to hundreds of times. The branching of an axon is called telodendria. And the axon terminals is also referred to as synaptic nubs. Same thing. Okay? Everybody got these terms? ASL. Now let's talk about that orange that's on the whiteboard over there. This is not true for all axons, but some of them will be covered by this neuroglial cell. Now in the PNS, it's called a Schwann cell, that's what's on the screen. But the newer name is neurolemmacyte. Here's the reality. All the axons are sitting in these neurolemmacytes. It just depends on, is it just sitting? Because this big cell here is the neurolemmacyte. All these orange things are the axons. If it's just sitting in it, it's considered unmyelinated. But if the neurolemmacyte wraps itself over and over and over and over again, then that axon is covered with phospholipid bilayer. You guys remember the membrane? It's mostly membrane. Just boom, boom, boom. Well, phospholipids, when it's concentrated like that, turns very waxy. Okay? And that would be a myelinated axon. So if the axon is myelinated, the outermost phospholipid bilayer is referred to as the neurolemma. That is the outermost. The thickness of all those multiple phospholipid bilayer is called the myelin sheath. But just know that the name of the cell that forms the myelin sheath is the neurolemmacyte, full name Schwann cell. And that's in the PNS. That was on one of those first slides of PNS, CNS. Look at that slide. Myelination in the PNS is neurolemmacyte. In the CNS, it's done by this different cell called an oligodendrocyte. Everybody now situated in what a neurolemmacyte is? Okay. The difference is that a neurolemmacyte will dedicate itself to one axon. It will just wrap itself around one axon, and it can cover about 1 millimeter of that axon. An oligodendrocyte, because it's in the CNS, there's a ton of axons, and the oligodendrocyte will branch and can myelinate several axons. Now, like I drew it on the whiteboard, there are gaps between neural lemmocytes, and those gaps are called neurofibril nodes. So this would be a neurofibril node. Covering this stuff this week in lab too. So the gaps between adjacent neural lemmocytes is called neurofibril nodes. Old name node de Grandier. Everybody see that? And again, in the CNS, because there are so many axons, oligodendrocyte will branch and can myelinate several axons in the CNS. But a neural lemmocyte will dedicate itself to one axon. So the neural lemmocyte is the faithful spouse, and the oligodendrocyte is the whole.
What is the horn? Is it still only wrapping it around one, even though it's doing multiple?
Yeah, it's going to wrap itself over and over and over again, but it'll cover, like in this case, 3.
But they're not clustered, it's still just one, in both cases.
Well, yeah, this arm is only doing 1. This arm is only doing 1. This arm is doing 1, but this one cell is doing 3. It's doing multiple axons. Any questions? Is anybody curious at all to what saltatory conduction means?